Hi everyone, my name is Sarah, and in this video we're going to be discussing cellular respiration, including the total ATP that we get from this process in the body. This is just going to be a brief overview. So the reactions of cellular respiration can be grouped into 4 steps. Step 1, glycolysis. Step 2, transformation, or as we'll discuss, the conversion of pyruvate to acetyl-CoA. Step 3, the citric acid cycle, and Step 4, electron transport chain, or oxidative phosphorylation. So Step 1, glycolysis. This is going to take place in the cytoplasm. Enzymes are going to split 1 molecule of glucose into 2 molecules of pyruvate, and this is facilitated by 3 main enzymes listed there: hexokinase, phosphofructokinase, and pyruvate kinase. And those are kind of diagrammed over there to the right about at what different points those take place. Energy to split that glucose molecule is provided by 2 molecules of ATP, and as glycolysis proceeds, energy is released. That energy is used to make 4 molecules of ATP. So this is going to give us a net gain of 2 ATP, again, as we produced 4 molecules, but we needed to use 2 molecules to split that glucose molecule, giving us those 2 as our net gain. And during this point, high energy electrons are also transferred to molecules of NAD+, which produce 2 molecules of NADH, another energy carrying molecule. So far we have those initial 2 ATP molecules, and then we have 2 molecules of NADH. And so at this point, again, those 2 molecules, we're going to say can each carry about 1.5 ATP, which is going to give us 3. So kind of at this stage, it can kind of be said that we have a total of 5 molecules of ATP so far. Step 2 is going to be the transformation of pyruvate to acetyl-CoA. This is just a short step in between glycolysis and the citric acid cycle. The 3-carbon pyruvate molecule made in glycolysis is going to lose a carbon, which will produce a 2-carbon molecule known as acetyl-CoA. This is diagrammed over there to the right, and you can also see that a carbon is removed and takes 2 oxygens from pyruvate with us, which is going to result in CO2 exiting the body. And 2 molecules of NADH are also going to be produced during the oxidative decarboxylation of pyruvate. And this whole transformation is going to be facilitated by what's known as the pyruvate dehydrogenase complex. This is a total of 3 enzymes listed there that are hard to pronounce, and they're cofactors. They each have different ones, and there's a total of 5. So far, again, we have what can be considered the 5 ATP from glycolysis. And now in pyruvate, we have 2 more molecules of NADH produced. 2 molecules times the approximate 2.5 molecules of ATP gives us a total of 5 in this step. So far we are at 10. Step 3 is the citric acid cycle, and it's important to list a few of the key players in the citric acid cycle, which are the energy shuttles. We've discussed NADH a little bit so far, but again, this is an energy shuttle which delivers the high energy electrons to the electron transport chain, where they will eventually power the production of 2 to 3 ATP molecules each, which is where it gives us that approximate number of about 2.5. And when this electron shuttle is not carrying high energy electrons, meaning it has been oxidized, it is left with a positive charge and is called NAD+. Then we have the other energy shuttle of FADH2. This, again, is another energy shuttle that carries high energy electrons to the electron transport chain, where they ultimately drive production of about 1 to 2 ATP molecules each, which is where we get that approximate 1.5 that we're going to be using for our calculations. And additionally, the oxidized form of FADH2 is FAD, and that happens just like NADH. So the citric acid cycle is called a cycle because the starting molecule, oxaloacetate, which has 4 carbons, is regenerated at the end of the cycle, so it kind of continues in that way. Energy can be captured from this cycle because several of the steps are energetically favorable. And then the release energy is captured as the electron shuttles, NAD+ and FAD, are reduced back to NADH and FADH2, shown there, as you can see them branching off of that cycle, NAD plus being oxidized to, or reduced, sorry, to NADH, shown there in the purple, and FAD being reduced to FADH2, shown there in the green. And it's important to note as well that the acetyl-CoA that was produced from our previous conversion from pyruvate is what is feeding into this cycle. And our final step, step 4, is oxidative phosphorylation, which takes place in the electron transport chain. During oxidative phosphorylation, NADH and FADH2 are transported to the electron transport chain, where their high energy electrons are going to be what's ultimately going to drive the synthesis of ATP. The two mobile electron carriers shuttle electrons between these complexes, coenzyme Q and cytochrome C, and 3 of what's going to be a total of 4 complexes translocate protons across the inner mitochondrial membrane, which are going to generate the transmembrane electrochemical gradient. This is summed up really nicely, and I think that picture is shown there to the right. So again, there's going to be a total of 4 of those inner mitochondrial membrane protein complexes. Complex 1, 2, 3, and 4 all have names listed there. In complex 1, NADH is going to be oxidized to NAD+. In complex 2, FADH2 is going to be oxidized to FAD. And in complex 4, electrons are going to be neutralized into water by oxygen. And then in a final chamber, not a part of one of the complexes, but in a separate one at the end, is called ATP synthase. At that point, ADP is going to combine with a phosphate to produce ATP, so that is where ATP is actually produced and released. And it's important to note as well, as shown in that picture before, that protons, those hydrogen ions, are being released into the cytoplasm during these processes, which give us that gradient. And each of the complexes is going to move electrons along a path within its structure, formed by a series of fixed redox-active prosthetic groups, or kind of cofactors, and those are listed there as well. So this has been a kind of a brief overview of those 4 steps and what that looks like, which gives us our total ATP yield. In that final step there, summed up as the acetyl-CoA feeding into the citric acid cycle, feeding into the electron transport chain, gives us, during all 3 of those processes, 2 ATP plus 6 more of those NADH, for a total of 15 ATP when you calculate their carrier abilities, and 2 FADH, which gives us 3 ATP, again, kind of showing there what they are capable of carrying. So this all total is going to give us approximately 30 ATP. This is not a set number. It can change a little bit, such as at the top there showing that the number of what NADH can be converted to regarding ATP can switch, even depending on the shuttle used, which could even go up and give us a total of 32 depending on that. But it's just a general approximation that we can always count on about 30 ATP being produced from our process of cellular respiration. I hope you learned a lot, and if you have any questions that might need a little bit of a further look into, this again has just been a very brief overview of the process of cellular respiration.